about the story that started in Dirk Hohmann's lab at Mount Sinai, and now I'm actually an instructor in Alice Campor's lab. So the work is a little bit similar, but also different. We're more focused on viral infections in chronic settings and also some tumor immunology. So I just wanted to very quickly start with like why we do need T-cell memory. Basically, it's a very, very short answer to that is to have more rapid and efficient protection when we get reinfected. So that basically when we start with like this very naive and like unexperienced SpongeBob after like an infection is cleared, when we encounter a second one, we are more protected and it can actually like, hit it out right away. It is surprising that we actually don't know that much yet about how CD8 T-cell memory is generated. So there has been a lot of discussions and multiple hypotheses how this actually evolves. But what we do know for now is that these like early memory precursor cells here in blue, they can be detected in among the pool of effector T cells relatively early on, and they have been identified by like a variety of markers, like previously mostly by the expression of CD127 and L-selectin, and then more recently people have started to look at TCF1 as the more defining factor of these precursor cells. And then what happens basically is that these precursors kind of evolve over time and basically de-differentiate and go back to what like looks more like a naive T cell in the end. And when you check the early memory T cell pool after infection, you would actually see that there is a different composition than at later time points. Like in the middle, like in the early phases, you usually have a lot of effector memory T cells, which look more differentiated and less TCMs. Whereas at the later time points, you would actually find a lot of TCMs and less TEMs. And these are all basically identifiable by a variety of markers. For example, CXCR1 or KLRG1 for the TEMs, and then more likely selectin and CD127 or CXCR3 for the TCM pool. And I also have to say that what I'm talking about is mostly about circulating memory T cells. It might be a little bit different for resident memory T cells. So overall, the evolution of T cell memory or CD8 T cell memory is associated with a lot of changes over time, and CD4 T cells can provide help during this process at various stages. And I think one of them we will hear about a little bit later. Most prominently is probably DC licensing via CD40 ligand and CD40 mechanisms, even though DCs can also directly activate naive T cells, especially in inflammatory or infectious settings where they actually can present intracellular pathogens. CD4 T cells also produce a lot of trophic factors like IL-2. They can also secrete more direct antiviral mediators like perforins. And then obviously they also help B cells by helping and assisting to generate antibodies that are helping in clearing pathogens. And then Tregs also play an important role, more likely at the later stages where they can actually limit the effector expansion of these effector CD8 T cells. So one question that we were interested in, as well as like a lot of other people, is actually what happens with this long-term memory CD8 T cell evolution when CD8 T cells are not primed with CD4 T help. And so a lot of people have studied this in the past, and this is where this term helpless memory actually comes from. And this is the observation that when CD8 T cells are primed in the absence of CD4 help, they usually don't create good memory. So like the purpose of memory, as I mentioned, is basically that you generate a better secondary effector response. And what people were observing was that when CD4 T cells were not present during this, excuse me, during this early effector stage or the priming stage, this did not happen. And very sorry. What is important is that basically there is two different scenarios actually. When you don't have CD4 T cell help at the very beginning, like when you encounter your first antigen, you usually also don't have a very good primary response. And that being the case, it is actually very surprising that when you do see a very good primary response during viral infections or bacterial infections, even when there are no CD4 T cells present during priming, you still end up with this like very bad memory. So multitude of like studies have been proposing mechanisms why this might be the case, transcription factors, cytokines, that the effector response is not programmed in the right way, or that CD4 T cells are needed to maintain the CD8 memory T cells over time. So basically, we really don't really know why this has actually happened. What we do know is that functional CD8 T cell memory actually usually requires CD4 T cell help. So when this project started out, like actually before I joined Dirk Hohmann's lab, like the main hypothesis was that because CD8 T cell memory is actually very dynamic and it evolves over time, as I showed you in this like first or second slide, helpless memory might actually not be a like, terminal defect and might be reversible. So to test this, we used the SNV system, which stands for lymphocytic choriomeningitis virus. And we use the acute version of this virus, which usually resolves within like 8 to 10 days after infection. And to probe this virus, or like the T cell response, we make use of a transgenic T cell line called P14. So these are mice that all express CD8 T cells that recognize a dominant epitope of the CMV glycoprotein called GP33. And we transfer like a very small number of these cells into naive mice that are then treated with isotype or an anti-CD4 depleting antibody. So the CD4 T cells are removed from the system, and they usually also don't return until like a month later. And then we infect these mice with the acute version of LCMV called LCMV Armstrong, and then basically just wait until the memory T cells develop. We can then look at the phenotype and the function of these cells, and we can also conduct adaptive transfer experiments. And what I mean by that is that we actually take out these P14 cells. We can recognize them by congeneric markers, so they are like different from the host T cells. And then we transfer them back into naive mice and can challenge these naive mice with like different pathogens that can be recognized by the P14 cells to study the recall response of these cells. So basically what we call secondary effector T cells. So using the system, we were able to recapitulate the like main or like signature memory defect of these helpless CD8 memory T cells, which is called the impaired recall expansion that people had been observed in the past. So here you can see that when these memory T cells coming from helpless mice are transferred into naive mice and then challenged with a listeria strain that also expresses the GP33 epitope, they usually do not mount a very good secondary response. And this is often the case when you re-challenge the naive mice with the original LCMV Armstrong. So when we look at the memory phenotype of these cells, like 60 days after infection, so these are the mice before we took out the cells and transferred them back into naive mouse for a re-challenge, we can see that the phenotype of these P14 memory T cells is very different in helpless mice. They usually express a lot of markers that are associated with like more effector memory phenotypes, so they're more differentiated. They express higher levels of CD43, KLRG1, CH3CR1, whereas like help memory T cells usually have higher levels of these like memory associated markers like CD27, L-selectin, CD127. So we then decided, because we're like trying to assess our hypothesis that this might be reversible, to follow these mice over time, and we tested we assessed the frequencies of memory P14 in helped and helpless mice in the blood, and you can see here that the levels, the circulating levels of these cells are relatively similar and maintained over time. There's not a huge difference. And then interestingly, when we look at the phenotype of these cells over time, we see what should be expected for the helped cells. This is here in blue, and then the red cells are always the helpless cells. And we can see that the help memory P14 actually evolved. Like the pool of these cells changes. They do upregulate CD27, L-selectin, CD127, CD122 over time, but then they actually lose expression of CH3CR1, KLRG1, and CD43. And we do see a similar like maturation kinetic for the helpless memory P14 pool, but it is delayed. So like you see this widening gap between the blue and the red line. And then also interestingly, you see like this widening standard deviation in the helpless memory T cell pool. So like what we concluded from this was basically that helpless memory T cells do mature, but they do undergo these like phenotypic maturation at a very delayed pace. So to then, like in principle, probe the potential of these helpless memory T cells to actually become good memory T cells, we sorted out helped and helpless P14s at the early memory stage around 40 days after infection. And we sorted different subsets of these memory T cells, TCMs, which are defined by L-selectin high and CD127 high expression. And then we have slightly more differentiated cells, which we call TM, based on this publication by Müller et al. In PNAS in 2022. So these cells basically still do have high levels of CD127, but low L-selectin. And then we have these like more like really terminally differentiated effector memory T cells that are double negative for both markers. And then we label these subsets with CTV, which is basically to track these cells over time and see how they dilute also, and then transfer them into naive recipients. So like helped or helpless memory T cell subsets go into these naive recipient mice. And then we analyze the transferred subsets 90 days later. And then I'm showing you here what these subsets looked like right after the sort before we put them back into the mice. So TCMs actually have a TCM phenotype, TMs are TMs, and TTMs are the TTMs, the terminally differentiated effector memory test based on CD127 and L-selectin. And then when we look at 90 days after transfer, we actually see that obviously what would be expected, the TCM stay TCM in the help mice, but they also do in the helped P14 memory T cells stay TCMs in the naive mice, but also the helpless P14 memory T cells stay TCMs in the helpless in the naive mice. So like this is not connected to the helpless P14 cell, but like they do actually have memory potential. And then interestingly, we also see that the TEM subsets do evolve over time. So like a lot of these cells that were originally selected negative start to upregulate it, and that's also the case for the helpless mice. And then more interestingly, actually also these terminally differentiated memory T cells are able of upregulating in both for the helped and the helpless subset expression of selecting CD127. And we think that is because like when we transfer them, about 30% of these helpless P14 actually also express TCF at that time. So like having TCF is probably a good thing. And when we look at the phenotype of memory and cells helped and helpless in the spleens 450 days after infection, which is about the time point that I showed you in this early kinetic, we actually see that there is no difference. So like all the markers that were different at these early stages between helped and helpless mice, they kind of converge. And when you look at this late memory stage, they look phenotypically very similar. Most of them have a matching TCM phenotype. Importantly, however, is that we also now see no differences in the functional compartment. So I'm not showing this, but they produce similar levels of cytokines. And then when you re-challenge these like aged helped and helpless P14 into naive recipients with LCMV or listeria that is expressing GP33, there is no discernible difference in terms of secondary effector expansion, which then led us to the conclusion that yes, in theory or in principle, helpless memory seems to be reversible because this defect is resolved over time. But what is interesting is that these helpless memory T cells kind of like mature at a delayed pace. Our next hypothesis was then that because like differences in the effector phase could probably like set the stage for the formation of helpless memory, maybe we see something at this time point. And so we followed the effector differentiation of helped and helpless P14 in mice that were infected with LCMV Armstrong from 5 to 8 days. And we did not really find a difference in terms of effector expansion. So they're very similar. And importantly, they're also very similar when you look at these like previously used like short-lived effector and memory precursor subsets. So there does not seem to be a huge difference when you just use these markers. We then also checked if these mice or if these cells are functionally different. We cannot see any differences in terms of cytokine production. And when we check for virus in these helpless mice, they actually also clear LCMV Armstrong within 8 to 10 days. So it does not seem that there is any difference at this point of the effector stage. To look at this in a more broadly way, or in a more broader way, we actually then conducted single-cell RNA-seq analysis, and we used P14s that were coming from helped or helpless mice at 7 days post-infection. So this is a little bit before the peak of the effector expansion in these mice. And then like found in principle on 5 different populations or clusters, which we called activated cycling 1, cycling 2, cytotoxic, or memory precursor, based on the signature markers that they express, and also comparing it to a like previously published data set. And then when we looked at the potential differentially expressed genes in these clusters between the helped and the helpless groups, we were actually not able to see any difference at all. So there is differences between each cluster, obviously, like the memory precursor cluster has more markers that are associated with memory usually, and then there's effector clusters that you have higher levels or higher mRNA levels of CX3CR1 or CXCR6, but there is no huge difference or substantial difference between the helped and the helpless clusters per se, and we also could not see any differences when we looked at cluster pseudobulk expression. So this was a little bit disappointing because like it didn't really provide an explanation why some of these like mechanisms and phenotypes that we were seeing were happening. But when we looked at the distribution of helped to helpless cells across these clusters, we could actually see differences. So showing you here, this is the TCF cluster basically, which we call memory precursor cluster. And then you have like several clusters that express high levels of granzyme A and granzyme B. And when you look at the distribution of helped to helpless P14 among these clusters, you can see that the helpless pool basically has higher cluster levels of or cluster expression of this activated cluster and of this memory precursor cluster, but then in on average, like the levels of cells or like the numbers that we found in the cycling clusters are a little bit lower. So it seemed that there was a like pre-distribution of like memory precursor cells in the helpless mice compared to the helped mice. And we were actually able to confirm this at the protein level, looking at granzyme B or granzyme A expression versus TCF1, where we confirmed that the helpless mice had higher levels of higher numbers of these memory precursor cells compared to the help mice. This is a little bit surprising, I would say, but like the summary that we got from this part of the project was that transcription and phenotypically effector cells, when they help us or help, they're very similar. And as I said, very surprising for us was that the effector differentiation was distinctly memory precursor bias. So like in theory, these cells would actually be better, should become better memory T cells, but that's obviously not the case. So we were assuming or like hypothesizing that it must be intrinsic defects or something in the memory programming that actually makes helpless effector T cells become worse and develop this helpless memory defect. So to test this, we did a couple of crisscross transfer experiments where we took helped or helpless effector cells and transferred them back into infection-matched helped or helpless recipients. So basically helped us go into help mice, but also go into helpless mice, and then helpless cells go into helped recipients and also helpless recipients. And these mice are infection matched, so all of them had been infected 8 days before. And then we look at the memory T cells later on. And then similar to what I've shown you before, we could not really see a difference in terms of the distribution of these cells or like the numbers of these cells that were maintained at relatively similar levels. But then what was surprising was when we looked at the phenotype of these cells, so this is the control here, helped cells go into a helped recipient, then a lot of these cells become TCMs, less so TEM, and very few actually, or like become these terminal effector memory T cells. And then in another control here, this is the helpless cells going into helpless mice. You see that most of them are actually these terminal subset. But when you transfer helpless P14s into a helped environment, they actually develop very normally and they look pretty much like this helped transfer control. Whereas when you transfer helped memory effector P14s into a helpless environment, they develop into helpless memory cells. So the environment seems to be very important in this context. And we tested it also by transferring the same cells into naive mice. So there is technically no inflammation, but also no antigen in these mice. And in that case, the help mice or the help P14 actually develop like completely normally, like they should, and the helpless P14 also develop normally, like they should, indicating that the post-effector environment is very important for the development of memory, helpless memory. So we were assuming then that maybe it is antigen, it could also be inflammation, but to test for the first, we actually used a very sensitive system transferring naive P14s that can act as a biosensor into mice helped or helpless that were infected 8 or 383 days before we transferred these cells. And then we looked at the proliferation of these naive P14s that have been labeled with CTV. And then you can see here that basically both in the helped and the helpless recipients, there is a little bit of proliferation going on actually beyond the point where virus is actually already cleared, but this ceases in the helped recipients. So there is no proliferation detectable after 20 or 26 days post-infection, whereas this continues pretty long and consistently in the helpless recipients. And we have individual mice even like 126 days after infection, where we are still able to see naive P14 proliferation. And to formally test that memory T cell subsets also would see antigen in these helpless recipients, we actually did a similar experiment as before. We sorted out these TCM, TM, and terminal effector memory T cell populations from helped mice, helped memory mice. They were CTV labeled, and then they were transferred back into helped or helpless LCV immune mice at an early memory time point and then a later memory time point. And you can see in this histogram plot here that in the helpless recipients, both the TCMs and the TEMs, again, these are helped cells that are transferred into helpless mice or helped recipients. They do proliferate quite a bit in the helpless compartment. The very terminal differentiated memory T cells, they usually do not proliferate good. But we only see this when we transfer them into helpless recipients. And then importantly, and I'm just going back here, you see that there's this huge portion of cells that have fully diluted CTV at this time point. So when you look at the phenotype before we transfer them in, they're all TCM or TEM. But when you look later on in respect to the proliferation that we observe, we can see that in the helpless recipients, this like fully diluted subset actually also phenotypically changes, and they all differentiate into more terminal subsets, which is specifically what might be happening in the helpless recipients over time. And when we look at a later time point, 60 days after transfer, we can see that actually the majority of these like helped TCM or TEM subsets develop into like more terminal subsets and helpless recipients, while this is not the case when we transfer them into helped recipients, where a lot of the cells retain or actually gain a TCM phenotype. And the same, and confirming basically the hypothesis that this could be antigen related. We do not see this when we transfer the same subsets into very aged, very old helpless LCMV immune recipients. Basically, the TEMs that we transfer at this time point mainly differentiate into like TCMs, basically. So to test if this is really antigen related, we made use of a virus that was made by Juan Carlos de la Torre and Beatriz Kubit. And basically this virus is a recombinant Armstrong strain that does express GP33, but in a mutated version so that the P14s cannot recognize it. And then we did co-transfer experiments where we took effector cells from help or helpless mice and we mixed them together at an equal ratio. And then we transferred them back into time point infection matched mice basically that were either infected with the wild type Armstrong. So this it's also a recombinant strain, but it expresses the GP33 epitope, or into mice in a help way or in a helpless way, or into mice that were helpless but infected with this mutated Armstrong that cannot be recognized by P14, or helpless mice that were infected with this mutant Armstrong that cannot be recognized. And then this is basically a control for this experiment where you can see that like similar to what I had shown you before, when you transfer help cells into helped recipients, obviously they stay helped. When you transfer them and when you transfer helpless P14s into a helped recipient, they also become helped and eventually look very similar to the helped counterparts. And when you transfer the same pooled population into a helped recipient, both of them obviously become helpless. But then when you transfer these combined cells, helped and helpless, into a mouse that does not present virus and is helped, it should look like very much like this helped phenotype. But when you transfer them into a mouse that was infected with the mutant virus in a helpless environment, basically, so these cells, these mice did not have CD4s when they were infected with this mutant virus. But since there is no GP33 that can be recognized by the P14s, they should also become helped. And that's actually the case for both the helped and the helpless P14s that are here transferred here. So when we were in revision, we were then actually asked, and we were thinking about this, like, does this also apply to other systems, not just the LCMV system? And we tested a Listeria, Vaccinia, and influenza. And actually, I'm just showing you the influenza data here, where we used a very similar approach. We transferred a small number of P14 into mice that were then depleted of CD4s or not, and then were infected with a virus that, a flu virus that also expresses GP33. You can see that they have very similar weight loss curves, so both of them recover. But when you look at the phenotype of the memory compartment, the help mice obviously have mostly a TCM phenotype, and very few cells that are like more differentiated TEM, whereas the helpless mice obviously in accordance with the previous LCMV data have less TCM and more TEM. And when you actually take these cells and perform a recall response experiment, we see the same as for the LCMV. Basically, for the LCMV system, basically helpless memory cells respond not as well as the help memory T cells to a secondary infection. And we are also able to confirm that we can detect prolonged viral antigen presentation in these helpless influenza-infected mice, even though this is not as pronounced as it was the case for the LCMV system. So at day 10 after infection, both of the groups actually clear virus. There's no virus detectable in the lungs of these mice. We still detect, similar to the LCMV system, a little bit of proliferation even in the helped mice. But then basically at 30 days after infection, there's no proliferation in the help compartment, whereas there is a lot or still a little bit of proliferation in the helpless mice that were infected without CD4s. So this is similar to the LCMV system, but it is probably not as pronounced. And then we tested as well if this could be caused by antigen that like kind of lingers on, like so the previous proliferation experiments kind of confirmed that already, but technically it could still also be inflammation. So we generated a virus which was made by Gabriel and Michael Swaptad, and they basically expresses GP33 or a mutated version of the GP33, and then we transferred effector P14 cells. These are helped, not helpless, they're all helped, into infection matched mice that were infected with the regular X31 that expresses GP33 and are helped, or the regular X31 that expresses GP33 but are helpless. And then a third group of mice where we transferred the cells onto helpless mice that were, but these mice were infected with the influenza virus that does not present a recognizable GP33 epitope. So we should, in theory, expect similar results as for the USNV system. And we actually do see that. So when we transfer the help cells into a helped environment, they all become TCMs over a short period of time. When we transfer them into a helpless post-effector environment, basically a lot of them look like they are helpless, but when we transfer them into a environment that is helpless but does not present antigen, they actually also become helped, and even maybe a little bit better than the original help T cells, confirming what we had seen previously in the ICMV system. We also were able to show very similar results for helpless infections with vaccinia virus or Listeria monocytogenes, which I'm not showing here right now. So in summary, what we were able to show in this paper, confirming this paper, was basically that, yes, absence of CD4 T cell help during acute infections can compromise the memory T cell pool and lead to like dysfunctional CD8 T cell memory. Basically, the system like predisposes the helpless T cells to have a higher composition of like memory precursor subsets. So we see this both in all of the systems that we looked at. But in general, all of these helpless memory T cells are effectively also undergoing memory maturation. It just takes them a little bit longer. And yes, like this helpless memory CD8 T cell defect that people have been observing over these like past decades seems to be a temporary effect, because in principle, it is reversible. And the main cause why we are seeing this in our systems is because we have prolonged antigen presentation, which is basically seeing stochastically or randomly by these evolving memory T cells that then basically start to differentiate back to a more terminal phenotype. And eventually, when you are at this like very terminal phenotype, you're probably not really coming back to being a good TCM in the end. So this is basically what we think, what is happening. And I think we also, we believe that this also has a lot of relevance today, like when you look at vaccination, but also other like diseases, autoimmune diseases, maybe even cancer, where T cell responses are primed without CD4 T cell help. And I think we will probably hear a little bit about this in a minute when Valentina is going to present. And with that, I'm just going to end this here. And I have a lot of people to thank, mostly Dirk, who started this together with Bennett, Kevin, who was a grad student in the lab, who helped with some of the experiments. I'm also very thankful to Alice, who is my current mentor and postdoc advisor, basically, and Etienne, Natalia, and Abhishek, who are all very supportive and great people to work with in the lab. Also have to point out Michael and Gabriel, who are like experts with influenza. Gabriel is like a virus magician. Basically, he can engineer any like recombinant virus strain that you're interested in. Same is probably also true for the Del Toro lab. They're like very, very good with all these recombinant Armstrong strains. And I'm also thankful to Vladimir, who did a lot of the bioinformatic analysis, Glaucia, and obviously also the Amit lab. Rafi actually had observed very similar things in the past. And this is actually also how we kind of decided to look more into this direction. And this work was supported by funding that I received from the German Research Foundation. And we also had NIH funding. And also, I'm very thankful to SciTech, because they give us money to actually conduct these single cell analysis. And the NIH Tetramer core is excellent when it comes to like providing monomers to use to track specific